Hello and welcome to this webinar on Multiomics for 5P medicine. Today I'm going to be discussing the concept of 5P medicine and how metabolomics and other omics are perfectly suited to help us advance medicine. After discussing 5P medicine, I'll look into specific combinations of metabolomics with other omics, especially genome-wide association studies or MGWAS, and also case studies with other omics. And we'll finish with a few words on how to perform multiomics data analysis. But let's begin with 5P medicine. To put it in context, I want to start by discussing chronic disease. In 2013, the WHO issued a global action plan for the prevention and control of non-communicable disease by 2020. As you can see on this slide, whether you look at diabetes, dementia, or cancers, these actions did not manage to curb the tide. By 2021, globally, 75% of non-pandemic related deaths were attributed to chronic or non-communicable disease. In 2023, Biocrates published a white paper diving into the origins of chronic disease, from classically metabolic disease such as metabolic syndrome and type 2 diabetes, to conditions with strong environmental determinants such as cancers, autoimmune disease, and neurodegenerative disease. In this document, we outlined the critical role of diet and its effects on the gut microbiome in the etiology of disease. This takes us to 5P medicine. This is a concept designed to combine steps in the application of molecular tools such as metabolomics to build the medicine of tomorrow. And whether it is preventive, predictive, or precision medicine, or for population-based medicine or participatory medicine, metabolomics is a powerful tool to develop solutions that take into account our environment in the study of health. And I think this is a great concept to help us tackle chronic disease. So I want to walk you through a few examples of how metabolomics can be used in 5P medicine. For example, given the role of diet and exposures in chronic disease, metabolomics is a perfect tool to devise strategies for the prevention of chronic disease, for example, with dietary interventions and monitoring of response through metabolite levels. Biomarker discovery is probably the best known use of metabolomics in predictive medicine, while metabotyping and stratification of individuals based on their metabolomic profile is a recognized tool for precision medicine. At the population level, metabolomics is the ultimate tool to study environmental variance, although it still remains the missing piece in many multiomics puzzles. And last but not least, advances in at-home sampling combined with quantitative and reproducible metabolomics that make the comparison to reference ranges possible are enabling patient-centric solutions through participatory medicine. One of the reasons why metabolomics is such a powerful tool for 5P medicine is its sensitivity to changes in the environment. This is also what makes metabolomics a great addition to multiomics approaches. The strong influence of our environment and lifestyle makes it crucial to study chronic disease with omic technologies that can sense the impact of our environment on our health. The scientific community has invested great effort in applying genomics to the study of chronic disease in the last 30 years. From this work, we've learned that only about 10% of the variants for any one of those chronic diseases can be attributed to genetics. The rest, that's governed by the way we live our lives. And for me, this means that the remaining 90% of variants are begging to be investigated with metabolomics, not only for cardiometabolic conditions, but also in oncology, in neurology, and other specialties. In fact, combining genomics and metabolomics and other omics is a great way to unlock extra information by bringing the pieces of the biological puzzle back together in multiomic studies. If you're interested to learn more about how to do this, I will get back to this towards the end of the presentation. But first, I want to look into the specific combination of metabolomics and genomics that combines information on genetic predisposition and phenotype. This type of multiomic analysis is also called mGWAS. In GWAS, statistics are applied to search for associations between gene variants and clinical phenotype, for example, a cancer diagnosis. These associations are generally weak. However, when adding metabolomics to the mix, strong associations can be found with both the clinical phenotype and the genetics. And this is what makes the combination of metabolomics together with GWAS particularly interesting. So when you build those strong associations, it becomes much easier to find new associations, to find new relevant mechanisms that could be involved or contributing to the disease. And when you already have the GWAS study measured, and you still have samples available that are compatible for metabolomic measurements, this can be a relatively small investment to combine metabolomics with the GWAS results. And because mGWAS is such a powerful use of metabolomics, this is one of the first topics that I wanted to discuss on my podcast, The Metabolomist, 3 years ago. So one of the first episodes we recorded was with Carsten Suhre, who is a German scientist working in Qatar, who was among the first people to combine these two omics together, and who really identified the power of metabolomics to find new associations and to strengthen existing associations. And what they also did from the very first paper where they combined those two omics is also to look not only at the associations with single metabolites, but also at the associations of gene variants with ratios of metabolites. And you see here in the table that while the association of each metabolite to a specific gene was not so strong, the association of the ratio of two here phosphatidylcholines was much more stronger with the same gene and allowed to identify that there was something in the variant here that was interesting for the study. So if you want to learn more about how to do mGWAS and what's the added value, I encourage you to scan the QR code on this slide or go to themetabolomist.com and listen to this episode with Carsten Suhre, where he explains a lot about the work that he's done combining those two omics and what's the added value there. And I want to finish this talk with a set of case studies. I'm going to go through them relatively quickly, but the point here is to illustrate the added value of combining metabolomics with other omics, and I hope that I can give you a taste for this with these case studies. The first one is a study where adding metabolomics has enabled finding new associations with exome-wide association study. So in the original study, combined with metabolomics, the scientists uncovered 85 variant metabolite associations, many of which were between metabolites and related enzymes or enzymes in related pathways. So this was very encouraging in terms of also interpretation of the results. The second example has to do with finding new pathways that can be of interest in the disease. So here we look at a combination of epigenetics and metabolomics. You can call that epimetabolomics. And here associations identified primary bile acid metabolism as a relevant pathway in the brains of Parkinson's disease patients. This was very interesting because studying the brain could identify things that you wouldn't necessarily find in people in a bit younger and earlier in the development of the disease. And this gave ideas to the scientists for future experiments on next steps to look for signs of disturbances in this pathway, for example, in the blood of living individuals with early stage Parkinson's disease. So this last case study has to do with metabotyping. Metabotyping is essentially using metabolic profiles of individuals to cluster them and group them into subgroups of the same disease. So in this paper, the disease in question was Alzheimer's disease, and the patients were stratified based on their metabotype, where already the scientists could see different profiles in acylcarnitine levels. Changes in RNAs and proteins were also measured and associated with these metabotypes, including with genes related to acylcarnitine metabolism and transport. So this also showed the power of using metabolomics to find new biomarkers and also of combining different omics to focus on specific pathways and specific markers in each pathway. So I hope that these examples encourage you to want to use metabolomics in your multiomics studies, but some people can be a bit intimidated by this data type and they're not used to it. So I want to say a few words about how to use metabolomics in multiomic data analysis. Essentially, there are two ways of analyzing multiomics. There's a very automated way using software that is not really tailored to your experiment, but can be very quick. And there's a very hands-on way, usually performed by bioinformatician, that is going to be tailored to your study, but can take a lot of time. And so at Biocrates, we offer a service that is in between these two and takes the best of both worlds, using the services of a company called ViewGene, where the bioinformaticians have developed semi-automated pipelines that are tailored to each type of omic. And the result is that you get your omic analyzed, each omic analyzed independently with the best possible pipeline for it, and then the integration of the different omics at the end. This concludes this webinar. Thank you for your attention. And I hope that these examples encourage you to want to perform multiomics and also to include metabolomics in your studies. So don't hesitate to reach out if you have any questions, and thank you for your attention.